Hello, my dear students. So, today I have come after long back with a new practical chapter. Okay, so the chapter name is the DNA isolation. So, all the students who have requested that if I will show you how the DNA extraction it will be done, basically the theoretical part, so it will be helpful for you. That's why I have come with this video how you will extract the DNA from the plant cell as well as the animal cell. But in this case or in this practical in biology subject, we have collected from the plant cell. So let's ahead to the class. DNA isolation is a process of purification of DNA from sample using a combination of physical and the chemical methods. The first isolation of DNA was done in 1869, which is very important, by the Frederick Mesker. Methods used to isolate DNA are dependent on the source, age, and size of the sample. In general, they aim to separate DNA present in the nucleus of the cell from other cellular components. Isolation of DNA is needed for genetic analysis, which is used for scientific as well as the medical or the forensic purposes. Scientists use DNA in a number of applications, such as introduction of DNA into the cells and the animals or plants, or for diagnostic purposes such as in the medicinal field. The latter application is the most common. Okay. On the other hand, forensic science needs to recover DNA for identification of individuals, for example, rapists, petty thieves, accident or war victims. Paternity determination and the plant or animal identification. Presence of proteins, lipids, polysaccharides and some other organic or inorganic compounds in the DNA perceptions or preparations can interfere with DNA analysis methods. They can also reduce the quality of DNA, leading to its shortage life. So these are all the introductory part of the DNA extraction. All of you know how the DNA it is important in our life, but here we will start the things. Okay, basically you have requested for the theoretical part. That's why I have shared with this video. Sources of DNA isolation are very diverse. Basically, it can be isolated from any living or the dead organism also. Common sources for DNA isolation include whole blood, hairs, palms, bones, nails, tissues, blood stains, then saliva, buccal means your cheek swabs, then epithelial cells, urine, paper curds used for sample collection, bacteria, animals, tissues or the plants. These are the all materials required for the DNA isolation. Okay. Stored samples can come from the archived tissue samples, frozen blood or the tissue, exhumed bones or the tissues, and ancient human, animals or the plant samples. Next is the procedure. There are 4 major procedures or the steps are hereby: that preparation of a cell extract, purification of DNA from cell extract, concentration of the DNA samples, measurement of purity of the DNA concentration. One by one we will be discussing here. To extract DNA from a tissue or cells of interest, the cells have to separated and the cell number or the membranes first you need to be disrupted. Then extraction buffer you need to carry forward. Chemicals such as EDTA, ethylene diamine tetraacetate you need to add for the removal of the magnesium ion, and obviously we need to add the SDS, which aids to disrupting the cell membranes by removing the lipids of the cell membranes are included in the extraction buffer. Otherwise the DNA it will be not visible. Having lysed the cells, the final step is the preparation of a cell extract is removal of insoluble cell debris. If any indigested particle or the waste particle it will be there, so we should remove the cell debris. Okay. Next we will go to purify the DNA from the cell extract. The most commonly used procedure are ethanol precipitation, phenol chloroform extraction and the mini column purification. These are the mainly most common procedure which we will be used for the DNA extraction. Precipitation. Ethanol precipitation usually by ice cold ethanol or isopropanol. Since DNA is insoluble in these alcohols, it will aggregate together giving a pellet upon centrifugation. So it is very much easy to extract or to do the ethanol precipitation. Phenol chloroform extraction. Phenol chloroform extraction in which phenol denatures proteins in the sample, because if in the cell we are disrupting the cell membrane, so we need to disrupt also the proteins, otherwise the DNA it is not visible for us. Okay, last, mini column purification. Mini column purification that relies on the fact that nucleic acid may bind to the solid phase. Okay, that is silica or other solid phase we can use depending on the pH. Obviously the cell pH it is varying, and the salt concentration of the buffer. Okay, next process is the concentration of DNA samples. The most frequently used methods of concentration is ethanol precipitation. So at the temperature of -20 degree or the less, absolute ethanol will efficiently precipitate polymeric nucleic acid. So it is very much careful step, so we need to do very much carefully. So these are the whole steps at a glance how you will extract the DNA. Okay, next, measurement of the purified DNA concentration. So after purification of all the steps what we have got, so we need to measure. DNA concentrations can be accurately measured by UV absorbance spectrometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA sample. Usually absorbance is measured at 260 nanometer. At this wavelength, an absorbance of 1.0 corresponds to 50 mg of the double-stranded DNA per milliliter. With a pure sample of DNA, the ratio of the absorbance is at 260 nanometer and 280 nanometer. Is 1 to 8. Okay, ratio of less than 1 to 8 indicate that the precipitation is contaminated with the different waste product, you can say, or it can be contaminated by the foreign particle. Okay, either with protein or the phenol. This is actually all the steps. You can take the sample from blood, you can take sample from the yeast, that is fungi, plant, animalia, bacteria. That means the bacteria, animals, plant, yeast, blood, they have different types of the cells, but the DNA proportion or the DNA, whatever the structure, it is quite same. Okay, based on the prokaryotic and the eukaryotic we can differentiate the structure, but actually whatever the DNA structure, it will be always diverse in case of this type of the animals. Okay, so stay tuned with this channel. I hope this video it will be helpful for you. If any queries or the questions is there, please don't forget to comment in the comment section, and please don't forget to subscribe my channel. If you are the new members, please subscribe my channel, press the bell icon, and don't forget to share the video with your classmates. Stay tuned. Thank you all of you. Have a nice day.